Hello lovelies, this is your video for the 2022 exam period based on the advanced information. This is for AQA, Separate Science, Higher Biology Paper 1. If you are not doing that exact exam, then look in the description down below to find the links to the other videos. So the chemistry videos, the combined science videos, the foundation videos. You can also get links to buy the t-shirts down there, which are awesome. Now, in this video and in the workbook that goes with it, what I've done is I've taken my big videos and my big workbooks and I've cut them up and I've chopped things around and I've moved them around to match the advanced information for this year. So we are going to start with what the exam board have told us are the major focus for this year's exams. Then in the second section we're going to be looking at the required practicals for this year's exams, and the last section is going to be the stuff that the exam board didn't list anywhere. So we know it's probably going to come up in linked questions, low tariff questions, maybe worth 1 or 2 marks. It is definitely worth still revising all of these. It does not include anything that the exam board has told us is not coming up in this year's exam period. If you are watching this the day before your first exam, then good luck. This might be your first big exam of this exam period. We are going to get through this. If you are watching this a few months before their exam, then good luck. Please don't stress over everything that's coming up. You and me, we are going to do this together. Our beautiful plant cell with a cell membrane that's responsible for determining which bits go in and out of the cell. A cell wall, important for structure. The vacuole, important for structure. The cytoplasm, where most of the reactions take place. The tiny little dots are the ribosomes, which are responsible for protein synthesis. The green bits are the chloroplasts. The pink ones are the mitochondria, where energy is produced. And then last but not least, we have our nucleus. Here we have our animal cell with our cell membrane, again controlling what goes in and out, our mitochondria, where energy is produced, ribosomes, which are responsible for protein synthesis, cytoplasm, where most of the reactions take place, and our nucleus, that's where the DNA is held, and that's a control centre of the cell. You'll notice there are several features of a plant cell that an animal cell doesn't share. For example, the cell wall, the vacuole, the chloroplasts. If you want to copy these pictures yourself, you can download them in the free revision guide from my website. Here we have our bacterial cell, which has its cell membrane controlling what goes in and out. The cytoplasm, where most of the reactions take place. The chromosome, the DNA, not in a nucleus. The flagella, which is used for locomotion. Ribosomes for protein synthesis, and then on the outside you have the cell wall. Even though you have to learn the structure of a typical plant cell or a typical animal cell, there isn't really a typical type of cell because there are a wide range of differentiated specialised cells. We can see here in our cross-section leaf it has lots of different types of cells in. Here we have a neuron, which looks very different to a muscle cell, which is going to look very different to a skin cell or very different to a set of cells in the gut. They're going to be specialised to do their jobs. So here we have villi, which give us long surface area. Here the cells are very tall to provide structure. Here the cells have a very long body so that the neurons can travel a long distance, and the muscle cells are going to stretch and contract. All cells start off looking the same, so they have your basic cell structure. And then various different genes will be turned on and turned off, and that's when it will start to specialise. That's when differentiation will take place, and it will grow this really, really long axon, or it will grow the villi, or it will turn into a leaf cell. Bacteria divide very, very quickly, from 1 into 2 into 4 into 8 into 16. A good bacteria, a happy bacteria, a bacteria that's got lots of nutrients and is happy with what it's doing, will divide roughly every 20 minutes. So that very, very quickly you'll go from one bacteria to millions of bacteria. So that you can become very ill from ingesting, from getting in a cut, from getting your skin, just a single bacteria, because they divide very, very rapidly. If you want to produce an uncontaminated culture of bacteria, moving your bacteria from one place to another, you first need to flame your inoculation loop so that it is red hot. This makes sure it kills everything that is on there. You need to make sure that you open your bottles near the flame so that no further contamination can get in there. Open the lid as little as possible, flaming the lid as you go. Work as quickly as possible to transfer the sample of bacteria that you've picked up into your uncontaminated broth, and working as quickly as possible so that you don't get any other bacterial contamination. You can then leave the sample at 37 degrees if you've got an incubator, or just leave it on the bench at 25 degrees for a few days and your bacteria will grow. I've done a much longer video explaining this, as you can see insert here. If you want to go and have a look at that, it's in the playlist with all of the other required practicals. When we are going to be looking at the effect of antibiotics or antiseptics on how bacteria grow, we need to make sure that our work area and our hands are clean, because even though these bacteria are relatively safe to use, we have to assume they're going to be pathogenic. You need to make sure you've labelled the underside, not the lid of the agar plate, and these plates will probably already be seeded for you by the technician. You can put your little filter paper discs on there, use forceps to do this, and then incubate them on 25 degrees for 48 hours. We can then measure the clear zones in 2 different directions. Here the clear zone is slightly hard to see, but hopefully if you look close enough you can see it. It's better if you measure the diameter, but in this case the only thing that I could do was to measure the radius because the clear zone was so large. When we're talking about diffusion, we are talking about things moving from a high concentration down the diffusion gradient to an area of low concentration. This could be things moving from an area inside a cell where they've been made to another area, or it could be things moving out of a cell. For example, it could be happening in the lungs. These are the alveoli, the air spaces, and this is the capillary travelling around it. These are very, very thin, walls only 1 cell thick, and carbon dioxide is going to diffuse from the blood into the lungs so that it can be breathed out, and oxygen is going to diffuse from the lungs into the blood so it can be taken around the body. All this can be in the gut. These are the villi of the gut. This is the gut cavity here, and you notice again they are 1 cell thick, and just like the alveoli have a very large surface area. We're going to get digested food moving from the gut cavity into the blood so that it can be taken around the rest of the body. So diffusion is the movement of gases or any particles that dissolved in solution moving down a concentration gradient from a high concentration to an area of low concentration. Osmosis is specifically the movement of water through a partially permeable membrane from an area of high water concentration to an area of low water concentration. So you'll notice this partially permeable membrane, the pores in it aren't large enough for the solute to move through, so the water is going to be the one that moves through here. This sort of thing can happen in root hair cells where we're looking at the uptake of water. Active transport again is a movement across a membrane, but it's from this time a low concentration to a high concentration against the concentration gradient. So our channel, our active transport channel, is going to pick up something that it wants. It is then going to move that through the channel to the other side. This could happen, for example, when we're talking about glucose in the gut or minerals in roots. Here we have an overview of our digestive system. The mouth, which is mechanically going to break down food. The salivary gland, which is going to produce amylase. The liver, which produces bile. Bile is something that emulsifies fats, so increases the surface area of fats, turning them from a big blob into a small blob and neutralises stomach acid. The gallbladder that stores bile. The small intestine that moves glucose, ions and other things into the blood and has a very large surface area. The stomach, which churns up food. The stomach acid, hydrochloric acid, kills bacteria and it provides an environment for proteases to work. Your pancreas, which produces enzymes. Your large intestine, which removes excess water, and your rectum and anus, which gets rid of waste food. There are a number of different enzymes in the digestive system that you need to be aware of. Lipase breaks down fats into fatty acids and glycerol. It is made in the pancreas and small intestine and works in the small intestine. Protease breaks down proteins into amino acids. It is made in the stomach, pancreas and small intestine and works in the stomach and small intestine. Amylase breaks down starch into sugars. It is made in the salivary glands, pancreas and small intestine, and it works in the mouth and small intestine. Amylase, protease and lipase are all enzymes and work with the lock and key mechanism. We have our enzyme, which has a very specifically shaped active site. So only one substrate or a couple of substrates are going to fit in there, the ones that have the complementary site. They're going to form an enzyme substrate complex. And then the enzyme is either going to break apart things or it is going to join together things. It is then going to release the products and then the enzyme is unchanged and can be used again. You need to know how temperature affects enzyme activity, and it is this kind of lopsided curve. When we have really, really low temperatures, there is not enough energy. At the peak, this is the optimal temperature, and then after the peak the enzymes get denatured, which means the links between them holding everything together are being destroyed. The enzyme is not killed. I know the temptation is to say this, but the correct term is denatured. Our curve for pH is much more symmetrical. We still have an optimal pH, but when it is too high or too low, the bonds aren't going to be in place. So the active site of the enzyme is going to be broken down. So again, it's going to be denatured. Here we have our respiratory system. Air goes in through the mouth or the nose, down into the trachea, which is also known as the windpipe, then into the bronchus, which is a branch of the trachea, into the bronchioli, which is a branch of the bronchus, and into little grape or cauliflower shaped alveoli. This is where gas exchange happens. And they have an incredibly large surface area. Your diaphragm moves up and down to bring air in and out. The heart pumps blood around the body. The intercostal muscles allow the rib cage to expand, and the ribs, the last part that makes up everything, protects the lungs. Here we have a cardiovascular system, and it is a double system. The blood gets pumped from the heart to the lungs, goes back to the heart, and then gets pumped around the rest of the body. If you see a picture of the heart, the first thing you do is write right and left on there. We have our vena cava, where the blood enters. It goes into the right atrium, down through a valve into the right ventricle. From the right ventricle, it goes up and to the lungs via the pulmonary artery. It comes back into the heart via the pulmonary vein, into the left atrium, into the left ventricle, and then is pumped to the rest of the body via the aorta. If you want to check you have the path of blood right, then we need to be looking at capital letters. It goes through the vena cava, the atrium, the ventricle, then the artery, back through the vein, into the atrium, to the ventricle, and then the aorta. So it goes vena cava, atrium, ventricle, artery, vein, atrium, ventricle, aorta. V-A-V-A-V-A. If you don't have that pattern, you've made a mistake somewhere. Other features of the heart that you need to know are here. These are valves. They will only allow blood to flow, and that this side has a much larger muscle than this side. The right side only needs to pump blood to the lungs, which aren't very far away, but this side has to pump blood to the rest of the body a much longer distance. The majority of the time, veins carry deoxygenated blood, apart from the pulmonary vein, which carries oxygenated blood back into the heart. And the majority of the time, arteries carry oxygenated blood, apart from the pulmonary vein, which carries deoxygenated blood from the heart to the lungs. If the heart isn't functioning properly, pacemakers, artificial pacemakers, can be introduced to help the heart keep time. Or if somebody has cardiovascular disease, then these tubes can get blocked up. Arteries have a very thick walls because they are carrying blood under high pressure, which means they have a thin lumen. That's the gap in the middle. Capillaries are very, very small. They are only one cell thick, or very, very thin, I should say. They're only one cell thick. This is to allow for diffusion. They generally go around in this kind of like mesh network around things like the gut, around the villi in the gut, around the alveoli in the lungs, so they have a large surface area. The veins carry deoxygenated blood. They carry it back to the heart, so they have valves, and they have thin walls and a thick lumen because they're carrying blood under low pressure. When we have cardiovascular disease, we have fatty deposits building up in the coronary arteries, the arteries around the heart. This can lead to the formation of blood clots. This blood clot can block an artery. This is going to restrict the oxygen to some cells. These cells are then going to die. If too many cells die, this can then lead to a heart attack. If so many cells die that the heart can't function properly or can't pump blood properly. Risk factors for this are going to be smoking, high blood pressure, or having too much salt or fat in your diet. Health is a complicated concept. It is going to be your overall state of physical and mental well-being. This is going to be affected by a number of things. It is going to be affected by your diet, exercise, community, whether you feel lonely, whether you have friends, and in part by your genes. Epidemiology studies are going to be looking at the levels of health and illness in a population. We need to do it in a wide population so we can look for different risk factors. For example, we can't ask people to eat a high-fat diet or to do lots of exercise or to drink lots, so we can compare them to other people who don't do those things or do those things. But there are people within a wide population that do those things already. So if we wanted to look at the effect of exercise on health, we could take our population, we could look at people that do lots of exercise, and compare them to people that didn't do any exercise. And because we have such a large population of people we're looking at, then we can compare the two groups. And we can follow these groups for years and years to see what the effects are going to be. Here we have a cross-section of a typical leaf. Our palisade mesophyll, where photosynthesis is going to take place. Cuticle, which is the waxy layer. Upper and lower epidermis, which cover the plant. Spongy mesophyll, which is a space for gas exchange, and the guard cell and stomata, which is where transpiration takes place. Inside the plant, we have the xylem and the phloem. The phloem is going to carry water. This is generally going to be in an upwards direction from the roots where it is collected to the leaves where it can be used for photosynthesis, and the phloem, which carries ions and food, and this is generally in a downwards direction from the leaf where food can be made in photosynthesis to the roots where it can be stored in, for example, potatoes. There are several factors that affect the rate of transpiration, and transpiration not only involves water moving out of the stomata but also moving up through the xylem. So if we have bright light, that is going to lead to more transpiration. More light means more photosynthesis, which means there's going to need to be more water brought up into the cell. If we have a high temperature, that is going to lead to more transpiration because the rate of reaction is going to happen faster. If we have high wind, that is going to lead to more transpiration, because wind is going to be brushing against the leaf or flowing against the leaf, moving things out of the way, so diffusion is going to play a part here. And if we have high humidity, this is going to lead to lower transpiration. Water is going to struggle to leave the leaf because there is a very high concentration of water. It's very humid outside. A pathogen is a microorganism that causes disease. For example, we can have viruses, bacteria, fungi, or protists. And these can be spread in a number of different ways. They can be spread in the air, for example, by coughing. They can be spread by touch. For example, if you have bacteria on your hands, or you have bacteria or virus on your hands and you touch a table and someone else then touches that same table. They can be spread through blood, sexual fluids, or they can be transferred via a vector like via a mosquito. Bacteria are going to make you feel ill because they produce a lot of toxins, so they'll give you things like food poisoning. Viruses will make you feel ill because when they reproduce, they cause massive cell death. Measles is a virus. It is spread by liquid droplets in the air or on surfaces. For example, if you sneeze or cough, then the liquid droplets that come out of your mouth and out of your nose go into the air, and if somebody breathes that in, they can get the virus. Or if that goes onto a piece of furniture, or like say the piece of train or bus that you're on, then you touch that and then you touch your mouth. That's how it can go in. the virus will quite happily sit around for a few hours waiting for someone else to infect. This is very common in children, and they're going to feel very ill. It's going to be a bit like having a bad cold or a bad cough. There's going to be coughing and sneezing, red eyes, high temperature, and a nasty rash. These days there is very little damage because there's a widespread vaccination program, the MMR, which prevents a lot of people getting it, and there are very few serious complications with measles. HIV is a virus. It can be spread in a number of ways, that is unprotected sex, sharing needles, childbirth, that's from mother to child, not just general childbirth, infected blood, breastfeeding from an infected mother. The implications are devastating for someone, although outcomes have rapidly improved recently due to the development of new drugs. So HIV attacks the white blood cells. White blood cells are an important part of your immune response. So if your white blood cells are being attacked, then you have little immune response. The damage is widespread, and HIV can develop into AIDS, where that's acquired immune deficiency response, which can lead to even the smallest infection having devastating consequences because you have no immunity against it. Tobacco mosaic virus is, unsurprisingly, a virus. It is spread from plant to plant by direct contact. The implications are a reduced level of chlorophyll, which is why you can see the mosaic pattern on the leaves. Some areas have a different level of chlorophyll than others. Now, if they have reduced chlorophyll, that's going to be reduced photosynthesis, which is going to lead to reduced sugars, meaning that there's going to be a lower yield from the plants, whether that's tobacco plants. This also affects tomato plants. So it's going to affect the commercial side of a business. Salmonella is caused by a bacteria. It's spread by eating infected foods. It lives in the gut of farm animals, so infected foods are going to be like things like eggs, meat, milk or poultry. However, it's very, very rare in the UK. We have eggs that have a little lion mark on them, which means they are salmonella free, and I don't think there's been a case of salmonella poisoning from eggs in years. The implications are going to be diarrhea, stomach cramps, vomiting and fever. Not very pleasant at all. And if severe dehydration sets in, then it can be life-threatening. Gonorrhea is a bacteria, which has a very long, complicated name. It is spread via contact with penile or vaginal fluid. It can also be passed from a mother to a newborn baby. The implications are a thick, green, smelly discharge from the penis or vagina, thoroughly unpleasant, pain and urinating, and bleeding. While the symptoms are thoroughly unpleasant, about 1 in 10 infected men and around half of infected women won't actually show any symptoms. Because the symptoms are so unpleasant, and because quite a large number of people don't actually show any symptoms, it is very, very important that you always wear a condom. Apart from being smelly and off-putting, the main damage here is going to be to your reputation, apart from if you're a newborn baby, when it can lead to blindness. Rose black spot is caused by a fungus. Fungus is spread by spores. You're going to see large areas of discoloration, which can actually lead to quite a lot of damage. It's going to be a loss of vigour. The plant's not going to be very happy because it won't be able to produce enough sugars. The leaves are going to appear black, they're going to appear yellow. They could all potentially fall off and then the plant might die. Malaria is a parasite. It is spread by female mosquitoes drinking your blood at night. It's not quite as sexy as Twilight made it out to be. The implications are going to be a high fever, sweats, and also chills, headache, vomiting, chest and muscle pains, and diarrhea. And this can be lethal in severe cases. The body is rather good at protecting itself against pathogens. The stomach is full of acid, which kills bacteria. Your respiratory system, your nose, your trachea, your bronchi are full of mucus and hairs which trap bacteria. Your skin acts as a barrier which stops things getting in. And your eyes have tears which wash them out clean. Your immune system is brilliantly clever at protecting you. It consumes pathogens, so your white blood cells will engulf, they will eat anything that they see as unfamiliar and dangerous, and then it will destroy it. They produce antitoxins to counteract the toxins that the bacteria produce, and they produce antibodies so that they can recognise pathogens faster. I imagine most of you have been vaccinated, or if you haven't, at least you've heard about vaccinations. Vaccinations are given generally to children or people that go on holiday to different places. And the childhood vaccination programme in the UK has prevented millions and millions of deaths and further millions and millions of serious illnesses. And in this country, it has wiped out a large number of debilitating diseases. It is very rare to hear of anyone getting polio these days in the UK because we are all vaccinated against it at a young age. The polio vaccine isn't too bad because they give it to you on a sugar cube, but it is quite painful taking your 8-week-old baby to be injected by the nurse. A vaccination is going to contain small amounts of dead or inactive pathogens. This allows your immune system to develop antibodies. So if you get infected with the disease at a later point, your body already has antibodies to it, it can recognise it, it knows it's pathogen, it knows how to deal with, and it can be dealt with quickly before you get ill. The advantages are that a large number of diseases has been wiped out. For example, nobody gets smallpox anymore or polio. And we have herd immunity, which means if a large percentage of the population are vaccinated against a disease, even the small percentage that have decided to not be vaccinated or can't be vaccinated for medical reasons are going to be protected as well, because the disease will find it very hard to spread. The disadvantages is that they don't always work. The polio vaccines, smallpox vaccines, are very, very efficient, but things like the flu vaccine doesn't always work. And it can be painful and there can be side effects. You may have heard about a controversy where somebody linked the MMR vaccine and autism. This is completely untrue. There is absolutely no link between these two. Here we have our lovely little mouse who's going to be vaccinated, and this is what's going to start the formation of antibodies. After a while, cells from the spleen of the mouse where the antibodies are formed are collected. We can take a known cell line, a cancerous cell line, myeloma cells, and we can fuse them together. After the antibodies and the cancer cell line have been fused together, we end up with a hybrid cell. These hybrid cells can be grown in culture in a laboratory until we have lots and lots of them. After they've grown up, the cells can be taken and the cells and the antibodies can be separated. The antibodies can then be used for various different things like pregnancy tests or cancer detection. Microscopy techniques have varied wildly over the time. From the very, very basic starts where you had your lenses and you had to use the focus to see what was going on. These are all generally hand done, very, very basics. To ones that you're probably more familiar with in school, which have slightly more sophisticated lenses, to the massive ones that I used to work on, electron microscopes, where they're all controlled by a computer. If you want to work out image height, object height, or magnification from an image you've taken from a microscope, the calculation is magnification equals image height over object height. I have my potato chips here all ready. What we need to do now is to dry them properly. We are not squashing and squeezing the potato chips. That's not what we're doing. We're just drying them to get the excess moisture. We're just drying them to get the excess moisture off them so that we are not going to affect the concentration of the solutions when we put them in. So just dabbing them gently, moving them around, making sure we get each side dabbed gently. And these will be made using a small potato chipper so that they are rectangular cuboid shape. Don't forget to get the ends. And then once we've done that, we can start weighing them. Here are my tiny pocket scales, which I guess I could take around anywhere with me. I've got the tissue paper lined up in front of here, so once I decide whether it's going to be 0, 0.2, 0.4, 0.6, I can put it in front of it so they don't get mixed up. I've got my iPad here to write the results table down, and we can get started. So that is 2.36, and that has gone down on the 0, so we can take that, just pop it in front of the 0 one over here so you don't get confused. Check that it's gone back to 0. It's always important your scales go back to 0 before you start measuring, or you're going to introduce an error. Once we've weighed them and recorded it, we can take our potato chips, drop them in to the tubes, make sure they are fully submerged. That wasn't planned, that was just completely unscripted. Just poke that down a bit. Make sure it is fully submerged. Now you can see these ones over here at the end in the 100% sucrose solution. These are actually floating a bit. If I poke it down, the same with this one. Oh, that's just a little bit stuck. These ones are definitely floating over here. So you can see a very clear difference between over here and over here. We'll start my timer. And we're just going to leave that for 20 minutes. Once these have been incubating, we can take them out of here, dry the potato chip again, weigh it again, and then pop the results in our results table. So you've just seen me do the experiment. Here is all of my data that I collected in the experiment. We can now work out the change in mass. So we are going to be doing final minus initial. It is really important to state whether it is going up or down in the change of mass, because we are going to need that when we're drawing the graph. Change in mass over initial mass times 100. So it's the percentage change from the initial that we are doing, not from the final, not from anything else. It is these numbers that we can use to plot our graph.
And that is done with the chemical iodine. You've also got the test for proteins. That's done by a chemical called Biuret solution. And you've got the test for lipids, done with ethanol and water. And then you've got the test for reducing sugars. Now, reducing sugars include things like glucose, but not sucrose. In the test for starch, you'd add a few drops of iodine, which is red-brown. It'll also sometimes appear as yellow-brown. So both answers are acceptable to the examiners, and it would change from red-brown to what we call blue-black. And if there was iodine present, the change from red-brown to blue-black would occur quite quickly. The food test for protein, also quite an easy one. You'd need to change the protein to a liquid. Generally, you do that by putting the sample in a test tube, putting a little bit of water with the sample and really shaking it vigorously so that any protein molecules would dissipate into the water, and then you could test it. Biuret solution would start off being a light blue colour and end up as lilac if protein was present. So you'd start off with light blue, and if protein was present, you'd have a lilac colour. The whole solution would turn lilac. Biuret is a combination solution made up of 5% potassium hydroxide and 1% copper sulfate, hence the light blue colour. The test for lipids, you'd start off having some form of fat, and now the test for lipids takes advantage of the fact that fats and oils are insoluble in water, so they don't dissolve, and fats and oils will dissolve in ethanol, which is a type of alcohol. So what you'd start off with is you'd put your lipids inside of a test tube of ethanol. Now ethanol is clear, and so you'd end up with a clear solution with your fats. So you'd shake them together with ethanol and you end up with a clear solution. Then you would pour that test tube into another test tube containing water. And if a cloudy emulsion formed, so we call it cloudy emulsion, so it's kind of a whitish emulsion. If a cloudy emulsion formed, fats are present. In the test for reducing sugars, we use a chemical called Benedict solution. And Benedict solution is usually a beautiful bright blue colour. And Benedict solution needs to be used in a particular way. You'd have to put the reducing sugar in solution and then add Benedict solution and then place the reducing sugar into a hot water bath. So you put it into a hot water bath and you just let it lie there for a little while. And within the hot water bath, the reducing sugar would cause the Benedict solution to change green, and then it would change orange. And if reducing sugars were present, it would change all the way from bright blue, so bright blue, through green to orange. Now sometimes we call this colour brick red, and orange is also acceptable. And it's very, very important to remember the water bath is entirely necessary to test for sugars.
DNA is a long strand of deoxyribonucleic acid, made up of lots of letters A's, T's, C's and G's. And these twist round into a double helix. This double helix is still ridiculously long, so it further twists round so that it's in a chromosome. And this chromosome is located in the nucleus of a cell. In mitosis, we go from one parent cell to two identical daughter cells. First thing that needs to happen is that the DNA in the nucleus needs to condense into chromosomes, and then they need to line up down the middle. Once they're all lined up down the middle and all the checks are taking place to make sure that chromosomes aren't going to go astray, they can start to be pulled apart to either end of the cell. New nuclei will form, and then they will separate into two identical daughter cells. Stem cells are fantastic things because they are things that have the potential to turn into any other type of cell. They have a number of different uses. For example, if you're treating Parkinson's disease, they can be used to grow new brain cells. If we're talking about brain or spinal injury, bone injuries, then they can be used to grow new bones to fill the gap. If we have organ failure, we can grow new organs or parts of organs instead of waiting and making someone wait on the incredibly long transplant waiting list. If we want to make stem cells, then we take a nuclei out of an egg cell. We take nuclei from the patient's cell and insert that into the empty egg. The egg can then start to develop into an embryo. From this embryo, the stem cells are then removed and stem cells are turned into new cells. This does come with quite a lot of controversy because human embryos are going to be created and then destroyed, and there are lots of religious objections to this, people just saying that life starts when embryos are created, and people that object to the destruction of embryos. You need to know the difference between a tissue, an organ, and an organ system. A tissue is one type of cell carrying out one function. An organ is made up from lots of different types of cells carrying out a joint function. And an organ system are a group of organs that work together to carry out a function. So our hierarchy is cells, tissues, organs, organ systems. For example, we can have muscle cells, which are part of muscle tissue, which together contract, which form part of the stomach, churning feed, and this forms part of the digestive system. You may have seen plants in the summer covered with thousands, millions of tiny little black or green aphids, devouring the plant as they go. They will suck all of the water, all the nutrients, all of the ions out of the plant, effectively killing it. However, the good news is that ladybirds and ants love to eat aphids, so this is a way that we can have natural pest control. You can go on the internet and you can order a box of ladybirds and you can use these to control the aphids in your garden. Photosynthesis is going to take water, carbon dioxide, and turn it into oxygen and glucose. We can take light and we can put it above the equation, but do not put it in the equation because it is not a reactant. It's just a condition that's needed. You also need to know the symbols for these. So water is H2O plus carbon dioxide, which is CO2, goes to oxygen, O2 plus glucose, which is C6H12O6. This needs to be balanced, but it's a nice easy one to balance because it is 6, 6, 6. So you can just remember that it's 666. And when you're writing out your formula, make sure your numbers are little and are in the correct place. Because if you write this, that's wrong, and you will lose the marks. In photosynthesis, we are taking energy from here, taking energy from light, and we are locking it up in glucose. This is an endothermic reaction. It takes in energy. There are certain requirements for photosynthesis. First of all, we are going to need chlorophyll. That is our green pigment in leaves. We're going to need water and carbon dioxide because they are our reactants, and then we're going to need sunlight. And the levels of these can greatly affect how much photosynthesis takes place. The rate of photosynthesis is going to depend on the percentage level of carbon dioxide. As the percentage level of carbon dioxide increases, so the rate of photosynthesis is going to increase, but only up to a point. After this point, there are going to be other limiting factors. Past this point, we need to increase something like the water, light, or the temperature if we want more photosynthesis to take place. We could easily switch this out to the percentage level of water and the graph would look the same. Light intensity is important for the rate of photosynthesis. When it's nighttime, when it is dark, we do not have lots of photosynthesis going on. As we get further through the day, as we get more light intensity, the rate of photosynthesis will increase until we get to a point where it is no longer the limiting factor and other things like the reactants or temperature need to be increased. After this point, we need to think about increasing other things. Now, even though the graph is flat here, it looks like it might have stopped. It hasn't. There is still a steady rate of photosynthesis. It's just not increasing as much as it was down here. It's just a steady rate. When plants are very, very cold, everything acts very, very slowly. Not a lot happens. It slowly increases until a nice point where the enzymes are happy and there's lots and lots of photosynthesis going on until it gets too hot and they start to be denatured, and then the rate will fall off very rapidly. So we have our rate of reaction increasing with temperature and our optimal temperature and our enzymes getting denatured. It's really important that you remember that the enzymes are denatured. They are not killed, they're denatured. The actual rate of photosynthesis that takes place is much more complicated than depending on just one thing. It's going to depend on lots of different things all at once. The glucose from photosynthesis is going to be stored as starch. The most obvious example of starch is going to be potatoes. For respiration, we are going to take glucose, add it to oxygen, and come out with water and carbon dioxide. You need to know the symbols for these. So oxygen is O2, water H2O, carbon dioxide CO2, and glucose C6H12O6. This needs to be balanced, but it's a nice easy one. 6, 6, 6. You have to make sure your numbers are the right size and in the right place. So these ones need to be little numbers and these ones need to be big numbers. Respiration is an exothermic reaction, which means energy is given out. The best example we can see of respiration is Screaming Jelly Baby demo, where we take potassium chlorate, that's our liquid oxygen, add in our glucose, that's our jelly baby, and you can see the massive amount of energy that comes off it. Anaerobic means without oxygen. So for anaerobic respiration, we take glucose and we turn it into energy and lactic acid. Not as much energy as aerobic respiration because the glucose isn't fully broken down. Anaerobic respiration can also take place in yeast. So yeast will take the glucose and will convert it into carbon dioxide and ethanol. Ethanol is used in drinks and cleaning products, and carbon dioxide is used for a variety of things, but when we talk about in the context of yeast, that is what's going to make your cakes or your bread rise. Metabolism is the rate that chemical reactions take place in your body. For example, glucose being turned into starch, cellulose or glycogen, fatty acids and glycerol being turned into lipids, amino acids being made into proteins, glucose and nitrite ions forming amino acids, proteins breaking down to form urea, and all of this is going to involve energy. The energy is going to come from respiration involving glucose. That's going to take place in the mitochondria of our cells. Amino acids, they're important for building proteins. Proteins make up all of our hormones, all of our enzymes. These are the bits that actually carry out all of the reactions within our body. Lipids are important for maintaining our cell structure and for storing energy. Plant diseases can be identified in a number of different ways. This could be due to discoloration of the leaves. So here we have the tobacco mosaic virus, where you can see the leaves going coloured, or they could be a black colour developing, as in rose black spots. The leaves could fall off. It could have a loss of vigour, which basically means it falls over and looks a bit pathetic. The flowers could either develop wrong or they could not develop at all, or it could die. However, a poorly looking plant doesn't necessarily have a disease, it might have an iron deficiency. If it has low nitrates, it is going to have poor growth plus yellow leaves. If there are low phosphates, it is going to have poor root growth plus discoloured leaves. Low potassium is going to lead to poor flower and fruit growth, and low magnesium is going to be yellow leaves. This crossover into chemistry. This is why your NPK fertilizers are important.